I'd like to welcome our next speaker, Frederick McCune, who will be sharing their talk, Leveraging Community Science for Large-Scale Monitoring of Insect Pollinators, Insights from Abeilles, citoyennes.
Perfect. Thank you. I will talk this afternoon about leveraging community science for large-scale monitoring of insect pollinators with insights from our project Abeilles citoyennes. So large-scale monitoring of insect is really needed regarding the global decline of pollinators currently. Large-scale monitoring program must follow the temporal and spatial trends of pollinator communities in the most cost-effective way while remaining scientifically rigorous. These these monitoring program must be simple, reproducible in space and time, cost-effective, and they must provide sufficient sample size to detect decline if they are occurring. To achieve these goals, many community science programs have emerged in the last decade. Community science is basically using observation from volunteers to collect the data. Most of these programs are, however, based on photography, which is a challenge when you study wild bees, because wild bees are challenging to identify to the species level, which is the level you need to assess community trends. Um, for example, just in Quebec, we have more than 300 known species of bees, and other than bumblebees, other species cannot really be identified with photography. One solution for this could be to use volunteers and a community science approach to collect the data or the specimens, coupled with a taxonomist expertise for identification. And this is what we are doing with our Abeilles Citoyennes project, which I will describe for the rest of the presentation. Just to begin with, Abeilles Citoyennes is the French for citizen bees. Um, our project has an agricultural focus. So we want to study pollinators mostly in the rural areas of the province of Quebec with three main objectives. The first one is to monitor those communities over time and space. We monitor wild bees, but also overflies, and we started this year monitoring soldier flies and bombilidae as well. We also use this data to answer some research questions, notably regarding the effect of landscape composition and structures on the pollinator communities. And we do a lot of education and outreach in this program. So we collect a lot from the citizens, from the volunteers, but we try to give back as well to agricultural producers and communities with outreach on pollinators. So to start the description of the project, I'll describe the team behind it. This project is based in Université Laval in Valérie Fournier's lab. It was coordinated by Anne Leboeuf for the first year of the project. Since Anne left the project, we split the coordination of the project between me, Sabrina, and Amélie. And we also have collaborators from the Montreal Insectarium and funding from the Ministry of Agriculture of the province. The project started in 2019 with a pilot year. So we basically tested our protocols and web tools with a small number of volunteers before starting for 2 real years of data collecting in 2020 and 2021. Then we took a break in 2022 and followed with a phase two of the project for the last 2 years. Um, Basically, how the project work is that we try to recreate volunteers to collect the bees for us that and then they are sent to the lab for identification. There are two kinds of volunteer we try to recreate. So first, agricultural producers, as our project is mostly on an agricultural focus. So we we want to see the effect of agriculture on pollinators, but also they are our main target audience for outreach. So we want to work with those producers. But also, like any individual or corporate participants can be involved in the project. For the site selection, we have sites in all the rural areas of the province. We excluded the cities of Montreal and Quebec that are the two largest cities of the province, where the communities have been well described already. Um, to be involved, volunteer must only have access to a site that is open, exposed to the sun with no overhanging vegetation and some bare ground so they can set up the trap. Our yearly maximum capacity regarding identification and sorting of the sample is about of 150 sites per year. And we have usually like 100 volunteers following these sites because some volunteer follow more than one site. Um, the way it works, we send them a sampling kit at the beginning of the season. It contains pen traps. So they receive nine pen traps, three of each color, yellow, blue, and white. And then they also have dish soap, an aquarium net, well-packed bags, recording sheets, prepaid return envelope, and all the protocols they need. They need to set the traps 5 times during the summer. So from May to September during 24 hours. Then they collect the insect from the traps with the aquarium nets. They put them with alcohol in the well-packed bags. And then they put everything in the prepaid envelope at the end of the season, and we receive them in the lab for identification. We developed a web tool, so the Abeilles citoyen website you can see here. It contains a Q&A, many resources and information on the project and the inscription form for volunteers. We also have that map of all the sites of the project where you can see the data, and all volunteer, they have their own page with their site information. So you here have an example from one of the site, and they also have access to all the data from the pollinators they collected. So they have the list of all their specimens, and they can filter them, for example, see the number of species for per genus or the number of specimens for genus and so on. Um, Here you have the map of all the sites from 2019 to 2023. So the sites from this summer are lacking on the map. We didn't process all the information yet. However, you can see the region number 9, which is the Côte-Nord, was the only region that was lacking volunteer in the project, but we have volunteers from this region this summer. So we now covered the whole province of Quebec. From 2019 to 2023, we had 161 volunteers collecting pollinators at 217 sites. If we take a look at some results for the same time period, so still excluding this summer as the specimens are still being processed in the lab, we collected 24,600 insects, so nearly 21,000 wild bees, more than 252 species, because we still have some genera to identify, and at least 33 genera. For overflies, we have 3,700 specimens from 39 genera and 86 species. We even recorded some at-risk species, notably bumblebees, so Bombus furvidus, Pennsylvanicus, and Terricola, were cuffed by some of our volunteers that give us hope that our monitoring program can work to follow even pollinators with declining and small population range. Regarding the outreach and education, we first do a webinar every fall with all the participants of the project. And during the summer, we also do many on-farms activities. We visited some of our volunteers. some agricultural producers doing activities under Farm Demonstration Day with advisory clubs and that kind of things. We also did activities for the general public, notably in national and provincial parks, as well as visiting some education institution for outreach with students, for example. We published a paper on this project. If you want to know more about it, I invite you to scan this code and check it out in Conservation Science and Practices, where we describe the perspective and insights, so more details about what I'm telling you today. In the last 2 years, some challenges emerged. One of those was recruiting agricultural producers. So it's not that producers are not interested in the project, they are usually really motivated. Um, However, they are really busy in the summer, so they get involved at the beginning, but never send back, never have time to collect the specimens and don't send them back at the end of the season. Some improvement for this was to work with agri-environmental advisory clubs to increase the number of sites. So these clubs, they visit many of their clients, many producers, and they can more easily do the protocols for us. So some clubs follow as much as 12 sites this summer for the project. Another change we made lastly was to get more activities for super volunteer. I think it's a common thing in citizen science project that you have super volunteer. People really engaged in all the activities of the project. And so we build a new protocol and send nets to those super volunteers so they can now catch bees differently and contribute even more to the project. Um, so thank you very much for listening to my presentation, and I will now take any questions as I think we still have some time for these.